Hello student, good morning. Myself is Dr. Satish Chand. I am working as an assistant professor in School of Agriculture. So today I will deliver my lectures on plant disease. So as you know, that means there is a number of the disease in a plant. So some of the fungal diseases, some of the bacterial diseases, some of the viral diseases, but today we discuss about the fungal disease, that is a blast disease of the rice, which is one of the most important disease of the rice. This disease caused by the fungus Pyricularia oryzae, and it is a classification of this fungus, that means name of the disease, blast disease of the rice, and causal organism Pyricularia oryzae, kingdom fungi, phylum Ascomycota, class Pseudomycetes, order Magniporthiales, family Pyricularia, and genus oryzae. And now how will identify, how will recognize this blast disease symptoms when we are sowing the seed in a nursery? That means the symptom is also visible in a nursery or as well as the transplanting rice field. So there is some peculiar symptoms, that means the blast is visible on a leaves, nodes and a neck. But you know, among all these three diseases, the neck blast, one of the most dangerous diseases of the rice. If we are talking about the symptoms, how will we identify, how will we recognize this is a blast symptoms of rice plant? So in the initial stage, we will observe there is some eye shape or a paddle shape spots are visible. In initial state, these spots are very minute and looking eye shapes, and a later stage when the plant become a very that means in later severe stage, these symptoms is visible from the distance as a paddle shape spores. And next one, this is a node blast. This is also very important disease of the rice. It is the second phase of the blast disease. So in the neck blast, generally the node of the plant are infected, and initial stage you have to observe there is some olive green and yellow green symptoms are appear on the node of the plant. But in the later stage, that from the neck it start the rootings and the finally plant down toward the soil, and this is a symptoms of the node blast. But when after some time when plant reach to the maturity, then this is the most dangerous stage comes in a rice field. It is called the neck blast. Why we are understand why we call it neck blast, because you know in locally that means neck is hang down, so that's why we are call it neck blast. Because in this case of the neck blast, there is no proper grain filling and there is the chaffy grains. If the initial stage disease take place, then there is no grain filling. But you know in later stage, if there is the panicles come out from the infected plant, then there is a partially grain filling of the rice spike. So this disease is called this stage is called one of the most dangerous stage of the rice. So these are some symptoms. On the basis of the symptom we can easily identify in a rice field. And now we discuss about the pathogen. So as I told to you, this is a pathogen. Pathogen is a fungus, and generally this pathogen is survive in a soil in form of the mycelium, plant debris or a spores. But here we discuss, that means the mycelium is a hyaline to olive, olivaceous and septate. That means hyaline is generally translucent, and septate, there are the two type of the mycelium. One is the septate, another one is a non-septate. So septate means there is a septa inside the mycelium, that's why we call it a septate mycelium. And these conidia are the pyriform. Pyriform means a pear shape. As that means you will be observe the fruit of the pear. So these conidia are looking as a pear shape. To attach to the border base by the helium, conidia are highlined to pale olive in green, usually 3 cell. As we discussed earlier, that means the conidia are a 3 cell. 3 cell, that means the cell is divided in a 3 portion. So that's why, and here you look, this is a conidia of the Pyricularia oryzae. These are pear shape, and this is a conidiophores. Generally the conidia are bears over the conidiophores. They are in a cluster shapes, and these conidias, after harvesting of the rice plant, the conidia survive, that means fungus is survive on the plant debris for a several months, and then there is a reproduction in a fungus, then the conidia are produced. There are the two type of the reproduction in a fungus, one is the sexual and second is the asexual reproduction. Student, as you isolate this fungus from the infected plants, so here you are looking at the mycelium, that means how we isolate this fungus. Generally from the infected plant we take that means we select the infected plant from the field and bring to the laboratory. First we rinse with the tap water, then we go for the isolation. The isolation is the process to segregate the pathogen from a plant and growing into the laboratory. We are using a media. Generally we are using a PDA media. PDA media is a potato dextrose agar media, which is generally used for cultivation of the fungal pathogen in a laboratory. So after isolation in a laminar air flow, that means this is a fungal mycelium colony. And now we discuss about the mode of the infection, how the pathogen infect the plant. As I told you, that means the fungus is survive in a soil in form of the conidia, plant debris or a mycelium, where that means there is no crops. So over winter or over summering the fungus is survive in form of the conidia. So these are the conidia. Generally the conidia are growing and it's produced the germ tube. This is a germinating conidia and this one is the germ tube. And this germ tube tapping over the surface of the plant, and then conidia, then mean pathogen get entry inside the plant cell. And after taking entry in the plant cell, ultimately it become a systematic, that means the pathogen is spread inside the cell and finally the plant got infected by the pathogen. So this is a cycle of the, that means this is a mechanism of the disease take place inside the plant. And another, now we discuss about the disease cycle, how the pathogen complete their life cycle. It is survive for the long periods in the field, or that means the many years in a field. As I told to you and we discuss, that means in a rice field, when we are harvesting the rice crops from the field, then the infected plant debris remain in the field for several years, and the farmer plowing the plant debris and burning of the plant debris, then these fungus is survive inside the soil in form of the plant debris, in form of the mycelium, in form of the spores. So here you are looking, these are the conidia. The conidia are survive in the soil. From these conidia the germ tubes comes and this conidia infect the sapling and the new saplings of the rice plant. And if the plant got infected by the pathogen, then there is a secondary infection take place. Secondary infection is generally take place by the transmitted of the spore from the infected plant to the healthy plant. But here we are talking about the primary inoculum. Primary inoculum is only mycelium, conidia, and as well as the volunteer plant. Those volunteer plant means those plant generally they are growing itself. It called the volunteer plant because in the absence of the main host crop, the fungus is also survive on a collateral plant. From the collateral plant, if the condition is the favorable, if the condition is the good for disease development, from the volunteer plant, these conidia are spread from the volunteer plant to the healthy plant, and then ultimately the healthy plant got infected by the blast pathogen. And as I told you, that means the secondary infection, generally that means the infection disease is generally depend on to the environmental condition. If there is a favorable environmental condition, because we are talking about the blast disease of the rice, generally this disease take place when the day temperature is up to 25 to 26 degrees Celsius, and the night temperature 20 that means the 15 to 20 degrees Celsius, and humidity 70% take place. So that's why at these conditions this blast disease pathogen generally infect the rice plant. So this is a disease cycle of this pathogen. And now we discuss about the last one is the management. So this is one of the most important techniques which we are using to control of the blast disease of the rice. That means generally we grow the resistant variety. There are the many more resistant varieties available in a market, such as a 15-0, that means 1509. So and there are some other CO47, IR20, AD36, 11, 21. These are the resistant variety. In a resistant variety, what do you mean by the resistant variety? That means the pathogen is unable to infect the rice plant. Generally, we are induce the resistant gene inside the crops, so the plant become a resistant against this particular pathogen, Pyricularia oryzae. And next one, avoid the cultivation highly susceptible variety. That the susceptible variety which is easily attacked, easily infested by the pathogen, these are called the susceptible variety. Because the coarse rice variety, such as a IRI 8, IRI 50, TKM6, these are the susceptible variety which is generally attacked by this Pyricularia oryzae pathogen. And next one, seed treatment. So the seed treatment, this is one of the most important strategy to management of the rice disease, and how we treat the seed before sowing of the seed in a nursery. We have to treat the seed with the fungicide. Fungicide, there is some fungicide which is generally used for treating of the seed. That fungicide is a chemical that control infection of the fungus. It is called the fungicide. So there is some specific fungicide like that, means Captan, Therum, Carbendazim, Tricycazole, and these fungicide is used for a seed treatment. And if you want to treat the seed, you have to take the 2 gram of the fungicide for the 1 kg of the seed. Just you take a 1 liter of the water and pour the 2 gram of the fungicide inside the water and properly rotate it, and then pouring the 1 kg of the seed and kept it for the 24 hours. And the next day put out the seed from the beakers or any buckets and then sowing in the field. So this is a process of the seed treatment. As I told to you, that means this have a many mode of the mechanism. It may be infect the crop from the seed, because the fungus is also survive in the seed. It may also infect the crop from the volunteer plant. So that's why you have to reduce or you have to eradicate also the volunteer plant from the field. In a rice field, so many weed plant is also growing along with the rice plants. So that's why you have to eradicate the volunteer plant by spraying some of the weedicide. And next one, we are talking about some of the bioagent, which is the most effective bioagent against this particular pathogen. So this is a Pseudomonas. Pseudomonas, it is a bioagent which is available in soil. We can easily isolate it in a laboratory and use against this fungus pathogen. How will you treat the seed with the Pseudomonas? Just take a 10 gram of the Pseudomonas for 1 kg of the seed and kept the seed over within a 24 hours, and next day use the seed for a sowing. And another one, spray the nursery with the Carbendazim, because I told to you this disease also visible in a nursery at the initially day of the rice plant. So how will you control it? And if there is a symptoms is visible on a rice plant, then you have to spray with the Carbendazim. This is a most important fungicide to control this Pyricularia disease. That means how much quantity you have to use, and 500 milligram per liter, and another one tricyclazole 300 gram 3 milligram per liter. If after doing all these practice or using all these methods, again the diseases appear in a field, so what will you do? That means generally in a severe condition or a epidemic conditions, you have to spray the Carbendazim or a tricyclazole at a 15-day intervals, and after repeating of 2 and 3 spray in a field, it is sure, that means it will be minimize the losses of the crops. And that we are talking about the how much loss due to this Pyricularia oryzae. In a severe condition, there is a 60% losses due to this fungus. Generally, these days a very resistant variety are developed by the plant breeders. So that's why this disease is not comes in a field. We are talking already in 1509 and 11021. These are the variety which become a very resistant, and there is a other method is also you can use to management of this disease. So that's why this is a disease of this rice crop, and so generally that means we are discuss about this important disease of the rice, and it is easily minimized, it is easily controlled by using a certain practice or certain control methods. There are many type of the control method. One is the mechanicals, second is the chemicals, third is the biologicals, fourth one is the chemicals. So that's why by you apply all these methods, we can easily control this blast disease of the rice. So as I told you, that means how to isolate the fungus from the infected plant. First, you have to visit the field and collect the samples. It is a very essential. You have to experience how to collect the sample from the field and how to identify, and it must be sure this is a blast disease of the rice. In some cases, I observed that means the students are not properly recognized this is a blast symptom. So how will you identify? I already told to you there are some specific type of the symptoms in case of the leaf blast. Generally the blast symptoms observed on a leaves, initially they are a minute spots, but after sometimes it become a very large and looking like a eye shape. And eye shape, generally here you look, this is a paddle shape, eye shape symptoms, and on the basis of these symptoms you can easily identify this is a blast disease of the rice, and this is a leaf blast. And second, node blast. Node blast is a moderately take place, because this is not a it is not necessary, that means the all three stages take place in a field, but the sometime rarely this stage come in the field, but it is also very mandatory. How will you identify, how will you detect this is a node blast? Because the fungus symptoms are looking very similar. In case of the Rhizoctonia solani, and the symptoms is also very similar in Rhizoctonia solani, because the symptom also appears on the sheath nodes. So it will very difficult to differentiate between the node blast and Rhizoctonia solani, sheath blight of the rice. But you have a well experience if you are continuously visit the field. So that's why we already motivate to you regularly. You have to visit the Sanskriti field, and then you observe this is a blast, this one is the rice, this is a nematodes, and this is a viral disease. So in case of the blast, how will we identify, how will we detect? There is a generally the symptoms appear on a rice plant nodes. In initial stage, these symptoms are very small and they are start rottings of the node, and the finally in severe condition there is the greenish mycelium is also appear on the infected parts. And the third one, this is the most dangerful stage of this rice, and due to this stage there is a various epidermics also observed or recorded in last 20 or 30 years. Because you know what the happen in case of the neck blast, generally lot of factors are influence this neck blast disease. Day temperature, night temperature, humidity also. If the temperature is a up to the 25 or 26 degrees Celsius in a day, along with the humidity 70%, then there is a more chance of this neck blast disease. And in this condition, and another factor is if the night temperature are going to dip, and it may be up to the 15 to 20 degrees Celsius, then there is a more chance, and the plant become a very susceptible and pathogen take place. So in this in case of the neck blast, so what the happen in the neck blast? Generally there are the two stage of the neck blast. One is the pre and post. If that pathogen take place emerging of the panicles, generally on the panicles there is a minute spots, and minute mycelium is also observed in a severe condition, and from the neck it will be bent down, and this is a major symptoms. Generally the local farmers of our area, or that means the jurisdiction of area of Sanskriti, call it gardan tod. That's why we are call it gardan tod, because you know the neck of the plant is bent down. In this case, there is no proper grain filling. There is no there is a partially grain filling, and the grain is also very cheapy, and there is a discoloration of the grain. So there is a lot of regions, and if there is a discoloration on a rice grain, what will happen there? Because you know you don't got the good price in the market, because you know if the crop is the quality of the crop is the good, so that's why it can easily sales. So these are the symptoms of the neck blast, and we already discussed about the pathogens. The pathogen is generally seed borne as well as the soil borne, because you know fungus is also survive in fact seeds or as well as a soil. So how can it survive in the soil seeds? When we are harvesting the infected crops and shattering or threshings, so generally the infected seeds mix with the healthy seeds, and the farmer store these seeds for the next crops cultivations. And if we are sowing the infected seeds, so what will be happen? Ultimately from the infected seeds the diseased plant will comes, and it will be transmitted the disease from one plant to another plant. So and next stage, it also survive in a soil. So how can it survive in the soil? It survive in the soil in form of the plant debris, because the farmer are harvesting the crops by hands, by machineries, then the residue living in the field, and it will be survive on the crop residue for within 3 years, 6 years, and when the favorable condition comes, then the spores of this fungus infected the crops. And third one is a volunteer plant. As you all know, the volunteer plant, there is some volunteer plant which are growing along with the rice crop. So these volunteer plant are the main source of the infection. There are the two type of the infection. One is the primary infection, another one is a secondary infection. Primary infection generally take place from the conidia. It may be, that means from the infected plant, the conidia survive in the soil. So the conidia which is survive in the soil start germination, and from these conidia germ tube come out and infected the plant. And the secondary infection is generally take place by the one, that means from the infected plant to healthy plant. Ultimately the plant got infected, and then the airborne conidia landed on a healthy plant. So this is called the secondary infection. It is very clear cut, that means what is the primary infection and what is the secondary infection. Primary infection generally initiation of the disease, it may be from the conidia, it may be from the volunteer plant, it may be a crop residue. And secondary infection is generally, that means the infection from the infected plant to healthy plant. And so these are the transmission of the disease. Now we discuss about the morphology of these pathogen. Generally we are talking about the conidia, and conidia is a source of the infection. It is also called the inoculum, and these conidia survive in the soil or over the plant debris for long periods. It may be 6 years, that means 3 to 5 years, and from these conidia it start the germinating and then infect the growing plant. And this is a conidiophores. So generally the conidia are growing over the conidiophores in a cluster. So that how will detach these conidia? So here you are looking, that means the conidias are separated from the conidiophores. It is looking like a pear shape. Pear shape, that means, as you know, the fruits. That means the shape of the conidia is a 3 cell. There is the 3 septations. So generally how can you identify this is a conidia of the Pyricularia oryzae? Because you know, from the infected plant you have to collect the plant infected leaves and make a slide and then go through the microscopy. Under the microscopy you have to observe the morphology of this fungus. Then you will be observed there is a numerous conidia inside the slide, and you can easily, if there is a 3 septations in a conidia, you can easily confirm this is a conidia of the Pyricularia oryzae. And now we discuss about the mycelium. So how will we identify, how will you check this is a mycelium of the Pyricularia oryzae? Because you know, basis of the morphology of the fungus and color of the fungus, we can identify this is a fungus of the Pyricularia oryzae and mycelium of Pyricularia oryzae. Initially, when we are isolated this Pyricularia oryzae, so you have to isolate over the PDA containing Petri plate. On the basis of the color, initially there is a brown color, but in a later stage it become a black. So this is a identification of this mycelium. And now we discuss about some management strategy. And as I told you, that means grow the resistant variety, and this is very essential because you know, in a market there is a number of the varieties available. So we easily purchase easily purchased from the any source of the market. So that's why it is recommended you have to select the variety, it should be a resistant, and you have to take the seed from the reliable source of the market. And another one, avoid the cultivation of highly susceptible. That means I already told to you, susceptible means those easily under the grip of the pathogen, they are called the susceptible variety. And remove and destroy the volunteer weed, because it is very essential and very compulsory when we are doing the rice cultivation, and along the rice crop some of the weed plant is also observed in the field. So what is the roles of the weed plant? Because you know the pathogens survive over the weed plant in absence of the host. So that's why it is a very essential to remove or eradicate the weed plant. Another one, seed treatment. Seed treatment, one of the best method to control of this disease. You have to treat the seed with the fungicide such as a Carbendazim, Therum, Mancozeb. You have to take 2 gram of the fungicide and 2 gram of the fungicide for the 1 kg of the seeds. And another one, foliar spray, which is very essential when the crop is at a maturity stage. You can also apply fungicide as a foliar spray to control of the rice disease. So that's why, and today we discuss about the one of the most important disease of the rice, that is a Pyricularia oryzae. So everyone knows that due to the Pyricularia oryzae, there is a many more epidemic already recorded in the history. In 1943, there is a epidemic in West Bengal due to the Pyricularia oryzae. So that's why it is recommended you have to take the seed from the reliable source which is disease, it should be a disease free. And where are you doing the rice cultivation? That means the field which is also should be neat and clean, and there should be a summer plowing, so that during the summer season the Pyricularia fungus comes over the upper surface of the soil, and it will become direct contact of the sunlight, and it will be eradicate from the field. So because you know, if you are talking about the losses of this fungus, generally there is a 60% losses due to this fungus, because in a severe condition it may be reach up to the 80 or 100% loss. So that's why we have to use the good management practice to control this Pyricularia blast disease of rice. Thank you.